All right, welcome back. Today we are diving deep, I mean really deep, into one of the most incredible factories on Earth. This isn't your typical assembly line. No, this one is a biological masterpiece, running non-stop 24/7. And the output? Well, it's just mind-boggling. Let's kick things off with a number that'll blow you away.
Yeah, you're seeing that right. 200 million. That's not a typo. That's roughly how many sperm a healthy guy can churn out in just one day. I mean, how is that even possible? How does the body manage that kind of insane high-volume manufacturing day in and day out? Well, we're about to find out. Let's go take a look.
So who's running this whole show? Well, pretty much everything, from building the factory in the first place to hitting those daily production quotas, is all managed by one single master hormone. You guessed it, testosterone. Think of it as the CEO, the factory foreman, and the main power switch all rolled into one. It's the boss.
Okay, time to start our tour. We're heading right onto the main production floor, the testes. Now, you might picture them as just these open sacs, but nope. They're actually super organized, divided up into about 250 little compartments, kind of like cubicles, called testicular lobules.
And inside each of those little compartments, you'll find anywhere from 1 to 4 super coiled up assembly lines. These are the seminiferous tubules. And here's the crazy part. You ready for this? If you could somehow unravel all the tubules in just one testis, they'd stretch out to about 250 meters. That's like 2.5 football fields of tubing packed into this tiny, tiny space. Wild, right?
So, nestled right in between all those coiled tubes, we find our first group of key employees, the Leydig cells. A good way to think about them? They're the factory's power plant. Their whole job is just to pump out that testosterone, the energy source that keeps those assembly lines humming along at full speed.
And then you've got the Sertoli cells. If the Leydig cells are the power plant, these guys are they're the support crew, the supervisors. Some scientists even call them nanny cells, which is perfect, really. They're literally attached to the developing sperm, feeding them, protecting them, and basically holding their hand through every single step of the process.
And this is where it all begins. Right at the base, lining the walls of those tubules, you've got the spermatogonia. These are your raw materials. They're the stem cells, the basic building blocks that every single one of those 200 million sperm will eventually come from.
Okay, so we've met the workers and seen the raw materials. Now, let's get to the really cool part, the assembly process. Because this isn't just about making copies of cells. Oh no. This is about a complete transformation, turning a simple, round cell into a sleek, specialized delivery vehicle. And it all happens in 2 main stages.
First up is a process called spermatogenesis. This is basically the multiplication phase. The starter cells divide, and crucially, they go through meiosis, which cuts their chromosome number in half. But after all that, you're still left with these simple little round cells. That's when the second process kicks in, spermiogenesis. And this is a total radical makeover. It's a physical redesign that sculpts that little round cell into the swimmer we all recognize.
So this whole remodeling job, spermatogenesis, happens in 4 main phases. First, the Golgi phase, where this little cap of enzymes, called the acrosome, starts to form over the nucleus. Think of it as a helmet. Next, in the cap phase, that helmet spreads out. Then comes the acrosome phase. This is the big one. The cell stretches out, a tail starts growing, and all the mitochondria, the cell's power packs, get bundled together to create an engine. And finally, the maturation phase. The cell jettisons all the extra baggage, all the excess cytoplasm, to become this lean, mean swimming machine.
And there you have it. The end result is it's a masterpiece of biological engineering, right? You've got the head packed with the genetic payload, a midpiece that's basically a motor, and a long tail for propulsion. It looks ready to go. But here's the twist. It's not. Not even close. At this point, it actually can't swim, and it definitely can't fertilize anything.
Yeah, believe it or not, these brand new sperm are basically duds. To actually become fully functional, they have to go through what you could call a finishing school. It's like a maturation maze where they'll get the final training they need for their big mission.
And they don't swim their way there either. They're still not mobile, remember? So they're just pushed along. A flow of fluid carries them through a whole series of ducts. First, the straight tubules, then into this crazy network called the rete testis, and out through the efferent ductules. All of this is just a transport system to get them to the next and maybe most important stop on their journey.
And that destination is a place called the epididymis. Now this thing looks small, but it's actually one single, incredibly long tube that's all coiled up on itself. How long? Try 6 meters. That's about 20 feet. To give you some perspective, that's taller than a giraffe. All coiled up in that tiny space. This is the training ground, and it's also the warehouse where the finished sperm are stored.
And this journey through the giraffe tube? It's not a quick one. We're talking about a slow 2 to 4 week trip. Yeah, weeks. This long transit time is absolutely essential. It's boot camp. It's where they finally learn to be sperm, and it's also a final quality control check before deployment.
So what exactly happens during this weeks-long boot camp? Well, a few critical things. For one, they finally learn how to swim, and not just wiggle, but swim with purpose, moving forward. Their acrosome, that enzyme helmet, gets some final tweaks. And maybe most importantly, their outer membrane gets coated with these special proteins. Think of it like putting the safety on a gun. It prevents them from freaking out and trying to fertilize something too early, which would be a huge waste of energy.
All right, our sperm are now fully trained and waiting in storage. So now we're at the final stage, preparing for launch. This is all about packaging and fueling. See, when it's go time, the sperm don't travel alone. They get mixed with a cocktail of fluids from a few accessory glands, and that whole mixture is what we call semen.
And this is where we get to bust a really common myth. If you ask most people what semen is, they'd say it's, well, sperm. But look at this. It's not even close. The sperm themselves make up less than 1% of the total volume. The other 99+%, that's all the fluid added by those accessory glands.
And each gland has a very specific job to do. The seminal vesicles add most of the liquid, about 70%, and it's packed with fructose, which is basically rocket fuel for the sperm. Then the prostate gland adds its own milky fluid, which has enzymes that are important later on. And finally, the tiny bulbourethral glands secrete a bit of mucus that helps lubricate the path. It's a team effort.
So, when the launch sequence is initiated, powerful muscles surrounding a tube called the ductus deferens, you might notice the vas deferens, contract like crazy. This propels the mature sperm out of storage in the epididymis, shoots them down the pipeline where they mix with all those fluids from the glands, and boom, semen is formed right at the moment of exit.
And just like that, the journey is complete. I mean, think about it. It starts with one simple stem cell, and through this incredible microscopic marathon of production, training, and packaging, you get millions of highly specialized swimmers, all prepped and ready for their one and only mission.
You know, when you step back and look at the whole thing, from that one little cell to this potential for new life, it's just one of the most complex, most elegant dances in all of biology. It really makes you wonder, doesn't it? What does this incredible microscopic marathon, with all its checks and balances, really tell us about the sheer precision and the amazing resilience of life itself?